Assalamu alaikum and welcome. In this tutorial we are going to discuss about structure and function of the cells. The first topic that we are going to discuss here, difference between animal and plant cells. You know that the animals cells are different from plant cells in the few organelles that are found in the animal cells and the few organelles that are not found in the plant cell, and few organelles that are present in plant cell but found not found in the animal cells. The organelles that are found in plant cell but not in animal cell are mostly flagellums. Flagellum help in motility, lysosome and centrioles. So the animals cell is built according to the lifestyle of the motility. It is here found the many large vacuoles, not a central vacuoles, and here the nucleus is in the center of the cells. But in plant cells, the nucleus is not in the center of cell because of the large vacuoles, large central vacuoles. Then there is no photosynthesis in the plant and animals because of the loss of chloroplast, and chloroplast are found in plant cells. Cell wall is the outermost boundary of cell wall of the plant's, but cell membrane is the outermost boundary of the cell of cell wall is the outermost boundary of plant cells, and cell membrane is the outermost boundary in animal cells. And there are centrioles, lysosome and flagella are not found in plant cells. So the next topic is plasma membrane. We will focus on the fluid mosaic model of the plasma membranes. And the structure of plasma membrane is composed of lipid and proteins. Lipid composed of 20 to 40% and protein composed of 60 to 80%, and small amount of carbohydrate is also present attached with the proteins or lipid forms. Then many biologists contributed to the structure of the membrane of the plasma membranes, and the unit membrane model and fluid mosaic model are the most important. Here is the unit membrane model, according to which it is composed of two lipid bilayers sandwiched between outer and the inner layer of the proteins, and central is hydrophobic zone, and outer and inner layer are the hydrophilic zone because of the protein structures. And then Fsangers, it was given. Then Singer given the model called as fluid mosaic model, according to which protein is not lipids are not sandwiched between protein particle, but proteins are embedded in lipid bilayer in a mosaic manner. Here it is not found between protein cells, but proteins are embedded in the lipid bilayer in mosaic manner. Here are the proteins, different type of protein, integral proteins and peripheral proteins. And here is the lipid bilayers, the structure composed of many type of the substances. Like here are the integral proteins that run away from the wall of the cell membranes, and here are the peripheral protein that do not run along the wall cell membranes. Here are the cholesterols are also found, which give the stability to the cell membranes. Here are it is actually it is composed of three phases. Here is extracellular phases, intramembranous phase, and cytoplasmic phase. And it is non-symmetrical because the things present here are not found in here, like the filaments of the cytoskeleton on the lower sides. Then there is cytoplasms, and on upper side we will see that the fiber of extracellular matrix, and that are attached. Here are the glycoproteins, and here are the glycolipids attached with the cell membranes. Here is the extracellular fluid. Here is the cytoplasms. Then we will study about here was the structure of the plasma membranes. And then we will study about the function of the plasma membrane, transport of material. One of the most important that is performed by cells. It acts as differentially permeable membrane and semi-permeable membranes, allowing some molecules to pass through it and some to not pass through it. The molecules that are most easily cross the plasma membranes are what type of molecules? They are lipid soluble molecule cross easily, and the small molecule like water molecule can also cross easily, and non-chargeable substances can also pass the cell membrane easily. Many small gas molecule pass easily, but ions which has charged particles have difficulty in crossing the membranes. Many substances which are not needed, we do not use energy at that, and they move by passive transport, and the substances that are needed and are found in against the concentration gradient can be taken in by active transport by using energy. Here we will also discuss the different type of the transport move across the cell membranes. The transport occur by using ATP in the form of energy, or the active transport, and transformations by and movement by which no ATP is used, are the passive transports like diffusions, osmosis are the passive transport, endocytosis, exocytosis, pinocytosis, phagocytosis, all are ATPs using mechanisms, and they are the active transports. In the many cells, the membrane helps to take material by folding in the form of vacuole, and it is called endocytosis. It is also performed by the cells. Many cells, it is also involved in the movement of the nerve impulse, and phagocytosis engulfment of the solid particle, pinocytosis engulfment of the liquid particles, and neurons nerve in cell membrane is also involved in the transmission of the nerve impulse. You know the neural, the basic structures. Then we'll come across the cell wall. The cell wall is the outermost boundary of the only found in certain type of the plants and the some fungi. It is composed of outer membrane, inner membranes, like primary wall, secondary wall, and middle lamella. What is the middle lamella? It is first to be formed between primary walls of the neighboring cells. Primary walls is the true wall, and is composed of cellulose, hemicellulose. Cellulose molecule are arranged in crisscross arrangement, like one fiber's layers is like one, and other fiber is at right angle to these particular type of fiber, and this type of arrangement of the cellulose molecule give strength to the primary wall. Then secondary wall, it is more stiff and provide more protections to the cell walls in inner surface, and comparatively thick and rigid. It is composed of inorganic salt, silica, waxes, cutin, lignin, prokaryotic cell like cellulase. Its strengthening material is peptidoglycans and murines. Here is the difference between prokaryotic cell wall and eukaryotic cell wall. And the fungi contains chitins, carbohydrate in their cell wall. We have also studied about it in the biomolecule sections, chitins and the polysaccharides. Then the cell wall is how much important it provide definite shape, keep the cell rigid. It does not act as a barrier to material passing through it is permeable, allow every type of the substances to pass through it. It is not giving a barrier to any type of the substances passing through it, so it is not acting as a semi-permeable membranes. Then we will discuss about cytoplasm. Cytoplasms is the inner substances, ground substances of the cells between the inside of the cell membranes. Here are the various organelles found. There is water molecules. There are the other extracellular substances. Likewise, insoluble, it composes insoluble waste and storage product. The soluble part is called cytosols, and gel part may be a cytogel proportions. Cytosol is 90% of waters. It is very important. It contains solution all the fundamental molecule of the life, where small molecules may form a true solutions, and a large molecule form colloidal solution. Colloidal solutions, there are maybe of two types: so like it is non-viscous, and gel like viscous. And peripheral part of the cytoplasms are likely gel. Peripheral part of the cytoplasms are gel, and central part of the cytoplasm may be of the sol, cytosol and cytogel. Two types of the cytosols are found in living organism. Most important function the cytoplasm is that it is a storehouse of the various chemicals. It is a site of metabolic processes like glycolysis. We have already studied in the bioenergetics that glycolysis part of the cell respiration take place particularly in the cytosol, and other part of the cellular respiration take place in the mitochondria. In living cells, the cytoplasms contain several cell organelles. Like what type of organelles are holded by the cytoplasm? All type of the cell organelles, including the nucleus, are holded by the cytoplasm. They are moving in a cytoplasm around the nucleus because of the large density of the nucleus. It is actually in the center of the cells, and all other organelles are moving around the cells and this type of the movement of the cytoplasm is called cyclosis. It is also responsible for the cyclosis movement of the water molecules. The most important function of the cytoplasms is to act as a storehouse of vital chemical. It also site for the certain metabolic process such as glycolysis. In living cell, cytoplasm contain organelles like endoplasmic reticulum, Golgi apparatus, nucleus, plastids, ribosomes, lysosomes, centrioles. Free-floating organelles like mitochondria, it is the example of free-floating organelles, move about in cytoplasm due to cytoplasmic streaming movement. We have already discussed about its. This is what type of the movement. It is the active mass movements, actively movement of the molecule in which mass substances of the substances move around the cells. Like here is the cell membrane, here the nucleus, all here is the different type of the organelles. They are moving in the counterclockwise or clockwise. In this way, these are the streaming movement of the cytoplasm. It is also called as cyclosis. Then endoplasmic reticulum is the specific type of the organelles around the. It is found around the nucleus. It is the labyrinth of the chains, and there will be container of the interconnected tubules around the cytoplasms. Uh, there may be connected with the nucleus, and the other end they may be connected to the membranous. These channels are called the endoplasmic reticulums. They are composed of vesicles, and vesicles are separated vesicle are cisternae and separated with each other by cisternal membranes. Now we will look about the morphological form of endoplasmic reticulum. There are two morphological forms of endoplasmic reticulum: rough endoplasmic reticulums with attached ribosomes, and here the ribosomes attached to the rough endoplasmic reticulums also form this type of the proteins, and so it is also help in the formation of protein, rough endoplasmic reticulums, and also maintain the shape of cells. Then smooth endoplasmic reticulums, which is without the ribosomes, and so it is not involved in the protein synthesis, but they also help in detoxify the harmful drugs and transmission of impulses. It is also used in the catabolism of lipids, metabolism of lipids, carbohydrates. It in a smooth endoplasmic reticulum play important role in the transport of material. Then endoplasmic reticulum provide mechanical support to cell. It is the function of both endoplasmic reticulum and smooth endoplasmic reticulums. But here these are the function of smooth endoplasmic reticulum, and productions of proteins are particular function of the rough endoplasmic reticulum. Here the nucleus, here the rough endoplasmic reticulum with attached ribosomes, here the smooth endoplasmic reticulums, and the proteins are then transferred to smooth endoplasmic reticulum. Then they are then transferred to the Golgi apparatus, where they are packed into these vesicles and moved out of the cells through exocytosis, or maybe used inside the living organism. Then we're going to discuss about ribosomes. Actually, it is involved in the production of the proteins. How they are protein, they are larger subunit, smaller subunit attached to messenger RNA, then transfer RNA attached to and there is the translation of the RNA in the form of the proteins. And we will 5 and 2 3 and the process is started, and ribosome is not included in syllabus, so we will learn only that the ribosome is the protein factory, and factory of ribosome are the nucleolus. Nucleus are the found inside the nucleus. Golgi apparatus. Golgi apparatus was discovered by Golgi in 1999. This is a very favorite MCQs of the MDCAT examiners, and so we have discussed it here. Then it is also composed of the sacs called as cisternae. Cisternae together form the Golgi complex. This complex interconnected system of tubules around the central stalk. It is very important line for examiners. Cisternae stalk are continually formed by fusion of vesicles. These vesicles form and combine with each other, and this combination of vesicles is actually the Golgi complex. And actually, they are coming from smooth endoplasmic reticulums. We have already discussed about it. The outer is the convex surface, it is a forming phase. Here the vesicles come to this from the smooth endoplasmic reticulums, and the maturing phase or the cis phase, trans phase or the concave phase is the forming phase. Sorry, transverse phase, where the Golgi apparatus, Golgi vesicles, and that are in the form of the protein enclosed in the vesicles are moved out. Outer concave surface is the forming phase, while the inner concave surface is the maturing phase. Here the Golgi apparatus, Golgi vesicles moves outward. It is near the cell membrane. Cisternae break into vesicle from the lateral. Here the cisternae are broken from the vesicles and move toward the cell membranes. Wall stalks, number of the vesicles moving from outer to inner phase. They are coming from endoplasmic reticulum, and they are going toward the cell membrane for export. Here we will study about different mechanisms. Here is the nucleus, where there is the transcriptions and formation of RNA. Then it entered into the here ribosome attached to the rough endoplasmic reticulum. Here you see there is the formation of the protein by translations. Then these proteins are particularly transferred to the smooth endoplasmic reticulum. Like here, smooth endoplasmic reticulums they are going to the Golgi vesicles, where they are packed inside the vesicles, and these vesicles can go to the plasma membrane, can used by the cells, used by the nucleus, can be formed by lysosomes. Lysosomes are also formed by the Golgi vesicles, and then they are maybe used inside the cell and outside the cells, like rough endoplasmic reticulum to the transport vesicle. From rough endoplasmic reticulum, they transport to the transport vesicle from Golgi apparatus, and the transport vesicles reaches the Golgi apparatus, and here the transport vesicles may be used inside or outside of the cells. There may be secretions. So the basic purposes of secretions and digestion, secretions and granules, and actually lysosomal enzymes are also formed by the Golgi vesicles. It is involved in the production of the lysosomal enzymes. It is involved in the productions of the particular type of the secretions. It involved in the many useful products that are converted into the final forms. And the most important function is that it take the proteins and attach them with the carbohydrate to form glycoproteins and lipid to form glycolipids. Here are the basic functions. Golgi complex is concerned with the cell secretions. Secretions produced from within the cells are ribosomes and then pass to the endoplasmic reticulum, then Golgi apparatus. These secretions are converted into finished product and packed inside the membrane before export. For example, pancreatic secretion like granules, these are also formed by the Golgi apparatus. And protein and enzyme which have to be transported out of the cells are also passed through the Golgi apparatus. It is the exporting machinery. Most important function of the apparatus is modify the protein by adding lipid and carbohydrate and converting into glycoprotein and glycolipids. These are the important foundations and the acrosomes of the sperm are also formed by Golgi apparatus, because this is also the covering around the enzymes, and it contains contain the hydrolytic enzyme. It is also like a lysosome. So lysosomal covering are also formed by the Golgi apparatus, and also the acrosome are also formed by the Golgi apparatus. Here we'll end our topic.